record, and it starts recording, when it starts recording. So, this should be solid.
Yeah, it seems pretty straightforward.
It's recording now?
Yeah.
Yeah.
Okay. You guys hear me? Greetings. Good morning. My name is Manzur, Moidunny. I'm actually not an undergrad exactly here. I'm a postbac student. I graduated from MSU with supply chain management and pursuing medical school. So it's been a blast being here in Wayne State. I know I've involved with some of you guys over here. Love the gym. It's been great chit-chatting with the rest of you outside the door. We find out if there's a quiz today or not. So the reason I wanted to talk today about the cell cycle, I'm taking cancer bio, which is bio 5640, which goes into depth into the cell cycle, and it's really involved in the cancer pathogenesis when people get cancer. So it really goes into depth how a lot of these mechanisms that we're going to go over today are involved in it, and how cancer kind of deregulates that pathway to cause tumors in people. So when I'm studying this, it shows me an appreciation, because we hear about cancer all the time through all these fundraisers and everything, but a lot of us don't know exactly how this works. So today, hopefully I'll show you how a lot of the little mechanisms are involved in the greater part of cancer and how they're deregulated for cancer to form. So let's get started. Overview of the cell cycle. So the cell cycle, essentially, like Dr. Zane said, is composed of interphase, and where it duplicates the chromosome or the genetic material. Then it goes into the M phase, where the cell actually divides completely. So the length of time that takes for those two processes is variable amongst a lot of cells, depending on what species it comes from, and the tissue in particular that those cells arise from. So for instance, you can see that embryos, it takes 30 minutes for them to divide. So every half hour, embryos are divided. They need to divide that fast, because once it's been fertilized, it needs to grow fast to mature into a greater entity. Intestinal epithelial cells, 12 hours. So epithelial cells, we're mammals. So in a day, they're dividing twice. And we can see that human liver cells, it takes a whole year for these to divide. So just keep that in mind every weekend when we're out, drinking Kool-Aid. That it takes a whole year for all those liver cells to replicate. Okay, so the cell cycle consists of four main phases. You guys may know these so far: the M phase, G1, the S, and the G2. The way that I'm going to go over them is we're going to start with G1, then S, then the G2 phase, then to the M phase. So the G1 phase, it's called the gap phase, G1, gap 1 phase. It's kind of like a transient phase where the cell's kind of just there, and it's kind of like observing its environment, and it has the decision to make, where it would kind of want to stay in that G1 phase, or it wants to proceed into the rest of the cell cycle. So that's a decision our cells make all the time, whether to stay there or proceed. We'll stay there for how long. And the cell can decide how long it wants to stay there, or it can actually completely step out of the G1 into the G0 phase. So from here, it can step out into the G0 phase, which is kind of associated with differentiation, when cells differentiate and no longer divide as correct. So it makes these cells post-mitotic, so they're no longer going to be divided. So that's kind of a lot of maybe neuronal cells in your spine, like some brain cells, kind of like that. They stop dividing and go to G0. So when these cells do go through this G1 phase, the next one is the S phase right here. this phase where it's where the chromosomal genetic material it's duplicated, so they have two sets of chromosomal material. And it goes into the G2 phase, this cell with two sets of genetic material, and then go into the M phase. Now the M phase consists of two parts, mitosis and cytokinesis. So what happens in mitosis is that now the cell has two sets of genetic material inside the nucleus. It starts to turn into two nucleuses. So essentially, it starts pulling the chromosomes apart into two different cells. The nucleus around the cells kind of starts disintegrating, and it pulls apart. So, and cytokinesis is when it actually finishes pulling apart and becomes two nucleus, the whole cell kind of becomes two nucleus. The whole cell converges, and the cell kind of like pinches closed between the cytoplasm. So it's made that entire in entirety it consists of the M phase mitosis, nuclear division, and cytokinesis, the cytoplasmic division. And I'm sure you guys have heard of mitosis, prophase, metaphase, anaphase, telophase. So that's not gone into depth here, but that's what consists of mitosis. Okay. Now the cycles, again, like they can't just go through the cell cycle just like that. Like there are some boundaries, checks and balances, and we call these checkpoints. And we're going to talk about three main checkpoints today. These checkpoints kind of control, essentially cell cycle control the cell cycle. So the G1 checkpoint, okay? This is one of the bigger checkpoints in my opinion. I'm going to go into it later. The G1 checkpoint is where, again, these cells are in G1 phase. They're transient, they're just kind of like in the G1 phase and kind of observing, analyzing its environment. Is this environment favorable for me to divide right now? Am I receiving the right signals? Are the right proteins sending me the signal to divide and go into the S phase, divide and completely become two daughter cells? So it also kind of like looks over the genome and stuff like that. So those two are the main things, environment and signals. So if that's okay, everything's the go-ahead, protein signaling go ahead, it goes into the S phase, where the nuclear material starts to divide, and there's two sets of genetic material, the chromosomes divide, and it goes into the mitosis when it divides, and the second checkpoint is at G2, before mitosis. Now this step is where after it's divided, the cell kind of like reviews the genetic material, the two sets of genetic material, and says, okay, they fully divided, for one, okay, it looks like they have divided, and it checks to see, has the genetic material been divided well, and are all the copies error-proof? Are there no mistakes? Is it okay? Is it safe for us to let this new material go into the daughter cells? So if that's to go ahead, everything's good, we go into mitosis, which is the first part of M phase. We go into mitosis, and when the cells, again, chromosomes, start to pull apart, kind of like in metaphase, where all the chromosomes are spindled up, and they're kind of split into two different halves of the same cell to turn into two cells. So at this checkpoint, the cell kind of looks it over. Okay, is everything equally aligned? So these cells have equal portions in each cell. Because if they don't, if one cell has more genetic material than the other, it's going to be bad for the cell. It can be lethal, essentially, for every cell has different amounts of chromosomes. So they make sure it's equally aligned. Each cell is getting different amount, the same amount of chromosomes in each one. So those are the three checkpoints. After that checkpoint, the mitosis checkpoint, the nucleus becomes two, and the cell becomes two completely different cells. Okay, so again, I want to point out something important right here. I'm going to come back at the very end. I'm going to come back to this slide because this slide holds important information about cancer. There's a point right here, kind of like the G1 checkpoint. It's called the R point. So the cancer takes advantage of this R point. The way it takes advantage of this R point is because cancer, the signaling molecules, the proteins that are involved all in this area, cancer takes advantage of them pushes it past this R point. And once it passes the R point is essentially like a roller coaster ride. Like you're going up, the R point's right here, and in cancer biology, it's like once you get past that R point, the rest of the cycle is like smooth sailing in a sense. Because up until the R point, your environment kind of determines. Your environment determines whether the cell is going to move forward or not. But after the R point, it's kind of like it's on autopilot. All the materials are already there. The cells know what to do after that. But it doesn't look into the environment. So I'm going to come back to this after explaining the mechanism so you guys will understand the parts involved. We're going to go over these mechanisms right now. So the cell cycle control system, essentially. Okay. So there are two main proteins involved in cell cycles. The control system is cyclin and cyclin-dependent kinases. You guys have any questions? You guys following?
Yeah, I have a question about the M phase.
Okay.
When you said how they separate equal amounts,
Yeah.
if they're not equal amounts, will it stop it entirely?
If they're not in equal amounts, it'll stop it. And if it's not able to repair itself, it'll probably, I'm guessing, it'll go into like apoptosis or senescence, where it just kind of like stops dividing. In a sense, the main point to take away is that that's the checkpoint for the material in our what we need to focus on. I think is that it checks to see if they are. There's more. You can go more in depth. There's different ways. That's what it does. Any other questions? I know I'm talking fast. Feel free to raise my hand. I'd be more than happy to answer questions. Okay, so where we're at, we have cyclins. Two main proteins, cyclins and CDKs. CDKs stand for cyclin-dependent kinases. So this cyclin-dependent kinase, you guys know what kinases are? It phosphorylates. Kinase phosphorylate other like proteins, other kind of things around cells. This kinase phosphorylates like the machinery. The machinery right here, this kinase phosphorylates the machinery here in order for the cell cycle to continue. And the cyclin right here, the cyclin determines where that kinase goes in that machinery. It takes it towards. So what happens is these cyclins and CDKs, when they're in the cell, they kind of come together, they bind, and then they're activated. They're activated by being phosphorylated, then they go ahead and phosphorylate the machinery. So what's key is here is the cyclin determines where it's phosphorylated, and the cyclin-dependent kinase does the phosphorylating. It does the directing right here, and cyclin binds that. Okay, so the accumulation of cyclins regulates the CDKs. So what we can see here is the cyclin, this is the cyclin concentration right here. It goes up during mitosis. This is mitosis. Interphase composed of G1, S, and G2. And this is the M phase. So what we can see here is the cyclin, this cyclin is going up during mitosis, coming down after mitosis, going up during mitosis, and coming down. So that's a cyclin level. So for the cyclin, it seems like they're changing. It's cycling in a sense, just like its name. So we can understand why they're called cyclins. They change according to when they're needed. So this particular cyclin is going up during mitosis, down during interphase, and back up. So what you can understand here is a big concept that these cyclins come when it needs to go to the next step, the particular cyclin. And CDKs, however, they don't change. We have a constant amount of CDKs in our system. So this right here, see how this barrier, this background gray area? So we can assume that that's a constant. This line, you can just like imagine that this is a constant line of CDKs. So the CDKs doesn't change, but the cyclin does. But the CDKs need the cyclin to be active. So the cyclin CDK activity goes up with cyclin concentration. So the CDK doesn't change, but the CDK activity does according to the cyclin. So it's all explained right here. Increasing cyclin concentration helps form the active cyclin CDK complex and drives it into M phase. The concentration of CDK component does not. Okay. Right here, so to learn all this stuff about cyclins and CDKs and everything, obviously you've got to learn about it, observe, and kind of do experiments and stuff to learn all this. So to do that, you got to observing cell cycles, and you can observe embryos, you can observe muscles, liver. But if you're observing liver, it takes a year for one cell cycle. So obviously you don't want to wait a whole year to study something. So scientists pick something fast to study. So this egg right here, it's a frog xenopus egg. After fertilization, it divides relatively fast, rapidly into eggs into many smaller pieces. So just imagine one egg fertilized, then it's continually dividing half, then into 4s, then into 8s, it's continually dividing. So you can observe a lot of cell cycles in that time. So it's like hardly any G1 or G2. It doesn't even stop the thing, and really fast, as repeated, as in M phases. For this, there's no need for new gene transcription. Everything's there, kind of like in a yolk kind of thing. They're all there. It just needs to divide. Like again, the cell doesn't grow at first. It's just like it comes in smaller and smaller pieces. So now we're going to go over this experiment, which is, in my opinion, kind of how like cyclins were discovered. Okay, they're experimenting with. They took an oocyte, and they took an egg. They took an egg in M phase. Okay, egg in M phase right here. And they took some of the cytoplasm out. So in M phase, what's going on again in M phase? In M phase, it's going through mitosis and cytokinesis. In mitosis, the chromosomes are essentially kind of pulled apart by the spindles and getting ready to separate. So that's kind of stuff that's going on in mitosis. So this cell, this egg that's going in mitosis, they took some of the cytoplasm out of it. And they put it inside this oocyte. They put the cytoplasm inside there. And what do you think they observed? They observed that this oocyte started going to M phase. The nucleus started dissolving, and the chromosomes started to be pulled apart. So I was like, what's going on? There's something going on in that cytoplasm that made this happen. So to test that, they did a control experiment. They essentially took an egg that was in interphase, not in M phase. They took an interphase, they took cytoplasm out, and they put it inside a different oocyte, just like the first one. And they realized nothing's happening if the cytoplasm is from interphase. Okay, so they realized there's something going on in the cell in M phase that creates this, the cell to go into M phase in mitosis, and the new one. So they call it the maturation promoting factor, MPF. It causes the cell to mature, essentially. They call it the MPF. So after that, they decided to observe it. Let's observe this MPF. And they realized, okay, wow, this MPF is going up during mitosis, down during interphase, up during mitosis, and down again. So, and there we go. Here's your MPF. M, you can call this the M cyclin during the M phase cyclin, and the CDK, they complex, and they're activated. They become activated, and they cause mitosis. The MPF activity rises rapidly just before the site of mitosis and falls rapidly to 0 towards the end of mitosis. So we've discovered a cycle. This MPF is working like a cyclin. So I guess, in my opinion, that's kind of like how they could have discovered it. So we're going to jump to how these cyclins and CDKs work. I mentioned that there's phosphorylation going on. So what is happening exactly is the cyclin CDK, when they come together, the cyclin, which is changing, when it becomes high, the cyclin CDKs come together, but at first they're not active right away. They're inactive. They have to be, first they're phosphorylated 3 times. 3 times. First time, they're phosphorylated at their activity site. Every enzyme kind of has a catalytic site. So it's phosphorylated at that catalytic site to be active. Then it's phosphorylated 2 more times. Right here it's only showing 1, but there should be actually 2 phosphoruses there. It's phosphorylated 2 more times to make it inactive. So we have 1 activated phosphorylation and 2 inactivated phosphorylation. So it's now an inactive CDK kinase. Okay? Then a phosphatase comes by. Who knows what a phosphatase is? Raise your hand. What is phosphatase?
It dephosphorylate.
It dephosphorylates, yes. So this phosphatase, activated protein phosphatase right here, it dephosphorylates two of the deactivated. Again, it shows only one here, but it should be two. Two deactivating phosphoruses, it deactivates it. So this becomes active. So now we have an active cycling CDK kinase. So now what happens is, there's different phases. The cyclin takes this active CDK kinase to the right part where it's supposed to go to that machinery. That particular protein machinery, it takes it